Hello everyone. Assalamu alaikum dear listeners. As we are studying the course of human physiology, after complete and detailed lectures and complete description about digestive system of humans and digestion and absorption of carbohydrates, now our today's lecture is all about digestion and absorption of proteins. Do you know what is a protein molecule? So let's introduce you about proteins. Protein is one of the most important substances in our body. Muscles, hair, eyes, organs, and many hormones and even enzymes. They are primarily made up of proteins. It also helps to repair and maintain body's tissues. However, not all protein is created equal, and there are things that you can do to help your body use it more efficiently. Protein is a very large nutrient that's made up of smaller substances which are called amino acids. They are key units of proteins. There are almost 20 amino acids, but our body can only make 11 of them. The other 9 amino acids they are called essential amino acids, and we can get them only from our diet. High quality protein sources such as meat, fish, eggs, and dairy products, they contain all 9 of the essential amino acids. So we don't need to take tablets or any supplements for these essential amino acids. Rather, we can take them from our daily routine diet if we take a balanced diet. There are also called whole proteins or complete proteins. Other protein sources such as nuts, beans, and seeds, they only contain some essential amino acids. However, if we combine some of these protein sources such as rice and beans to create a complete protein that contains all 9 essential amino acids. When a diet is rich in protein and the ingested amino acids exceeds the body's need for protein synthesis, the surplus protein is catabolized and amino acids they cannot be stored. This fact is contradictory to the carbohydrates. As we have already seen in our last lecture that that a surplus of carbohydrates that are not needed by our body, they are stored for later use. As compared to carbohydrates, proteins they are not stored in any form inside the body. Rather, they are catabolized to different products and byproducts. However, here one thing is important to note that proteins might be converted to uric acids, and uric acids can be stored inside the bodies, for example, in joints. Amino acids released go to the oxidative degradation during starvation or in uncontrolled diabetes mellitus when carbohydrates are either unavailable or are not properly utilized. Cellular proteins are used as fuels. So we can say that proteins are large biomolecules or macromolecules that consist of one or more long chains of amino acid residues. Intake of dietary protein is in the range of 50 to 100 gram per day, while about 30 to 100 gram per day of endogenous protein is derived from digestive enzymes and worn out cells. There are some important terms to know about proteins. The first of all is amino acid pool. Amino acid pool, they are free amino acids that are available throughout the body. They are captured in the tissues and fluids for use when needed. And the second one is protein turnover. Protein turnover, it is the approximately 300 grams of protein which is synthesized by the body each day, and 200 grams are made from recycled amino acids. In cell biology, protein turnover refers to the replacement of older proteins when they are broken down within the cell, and different types of proteins have very different turnover rates, while a balance between the protein synthesis and protein degradation is very important for good health and normal protein metabolism. As far as sources of proteins are concerned, we have two main sources from our diet, the exogenous source and endogenous source. About 30 to 100 gram per day of endogenous protein is derived from digestive enzymes and worn out cells of the digestive tract. The digestion and absorption of protein is very efficient in healthy human beings. While in terms of exogenous source, it ranges from 50 to 100 gram per day and is obtained from rather from animal source or from vegetable source. Animal sources that give us proteins, they include milk, eggs, meat, fish and liver. They are high rich diet proteins, rich protein diets, and the rich protein vegetable sources they are cereals, pulses, peas, beans and nuts. About 5 to 10 gram per day of proteins is lost through feces. Dietary proteins, they are denatured on cooking and are easily digested. Proteins are degraded by a class of enzymes, namely hydrolases, and specially cleaves the peptide bonds, which are known as peptidases. Here is the chemical formula of an amino acid that contains an amino group on the left side and a carboxylic group on the right side. R represents 2 to the methyl group. The type of amino acid depends upon the type of R group attached to the central carbon, while on the fourth side there is a hydrogen atom which is attached. The nature of this formula is very important as it has a carboxylic group, the acetic group and a amino group, that is the basic group. So overall the protein molecule is a zwitterine. Let's start digestion and absorption of proteins. The main digestion process of protein starts from small intestine. Digestion is a process which is essential for conversion of food into a small and simpler form. And we know that absorption, it's a process of transporting small molecules from the lumen of the gut or the small intestine into the bloodstream or lymphatic vessels. So the digestion, it takes place by two ways: the mechanical digestion and chemical digestion. The mechanical digestion, it occurs by mastication and swallowing, as we have already seen in case of carbohydrates. And chemical digestion, it happens to occur by with the help of enzymes. In small intestine, there are the digestion. Small intestine is primary site for digestion and absorption of food. Digestion occurs in the gastrointestinal lumen by secreted enzymes and on surface of enterocytes or by membrane-bound enzymes. Absorption occurs by simple diffusion. There are some processes which help for absorption of proteins or amino acids towards general bloodstream. That includes simple diffusion, facilitated diffusion, active transport, endocytosis, and paracellular transport. Surface area of small intestine is greatly increased by extensive folding, which are finger-like projections known as villi, and are also covered with microvilli. The purpose of villi and microvilli is to increase the surface area and hence to increase the rate of absorption across the intestinal wall. Digestion is hydrolysis of large and complex organic molecules of foodstuffs and mainly proteins here in this case towards smaller and preferably water-soluble molecules which can be easily absorbed by the gastrointestinal tract for utilization by the organisms. So digestion as well absorption, both they are complicated processes. Then comes the role of enzymes. Once the protein source reaches the stomach, HCl, the hydrochloric acid, and enzymes which are called proteases, they break down large protein molecules towards small. Hello everyone. Assalamu alaikum dear listeners. As we are studying the course of human physiology, after complete and detailed lectures and complete description about digestive system of humans, and digestion. Role of enzymes in digestion. Once a protein source reaches the stomach, HCl, the hydrochloric acid, and enzymes of stomach, they are known as proteases. That is, proteases, the name is given to these enzymes because of involved in the breakdown of proteins. So hence the name given proteases. They break down the proteins, the polypeptide chains, into smaller chains of amino acids. Amino acids, they are joined together by peptides which are broken down by proteases. From stomach, these smaller chains of amino acids, they move into the small intestine. As this happens, the pancreas releases enzymes. The pancreas releases enzymes and a bicarbonate buffer, the HCO3 buffer, that reduces the acidity of the digested food. This reduction allows more enzymes to work on further break down the amino acid chains into individual amino acids. That is the dipeptidases and tripeptidases towards free amino acids. Some common enzymes involved in this phase include trypsin or chymotrypsin, carboxypeptidases, and some other proteolytic enzymes. Peptidases. There are two types of peptidases. The peptidases that endo and exopeptidases. Endopeptidases that acts inside the core of protein and form small peptide fragments. While exopeptidases, they act from the amino terminal or carboxy terminal carboxy terminal ends of the protein. For example, if these enzymes, the exopeptidases, they act on the amino terminal, we say them as aminopeptidases, and if they act on towards the carboxy terminal, we call them carboxypeptidases. Proenzymes or zymogens. As all enzymes, they are secreted in the form in inactive forms, so they are known as zymogens. Actually, the peptidases are secreted in the inactive form in order to avoid itself from autophagy or autodigestion of the small intestine. When zymogens reach the site of action, they are activated automatically by the action of trypsin, another enzyme of small intestine. There are some proteases, as we have seen that there are two types of proteases, exopeptidases and endopeptidases. Endopeptidases, the enzymes that act on the internal bond of the polypeptide chain, while exopeptidases, they act upon the terminal bond of the polypeptide chain. Endopeptidases has nothing to do with the numbering of the polypeptide chain and the numbering of the bond on which they act, but exopeptidases, for example, there are some subclasses of exopeptidases. For example, aminopeptidases that act upon that break down polypeptide chain from the amino terminal of the polypeptide chain. They are known as aminopeptidases. Similarly, dipeptidyl peptidases that break down polypeptide chain from the amino terminal and produce two amino acid containing a short peptide chain. Similarly, tripeptidyl peptidases that act towards from the that act on polypeptide chain from the amino terminal and resulting in the formation of a short polypeptide chain containing three units. Then comes carboxypeptidases that help to break down polypeptide chains from carboxy terminal and separate carboxylic acid. Then comes peptidyl dipeptidases that breaks down polypeptide chain from carboxylic terminal and convert into a short polypeptide chain containing two amino acids. While dipeptidases that breaks down short polypeptide chains containing two amino acids, and tripeptidases that that break down three amino acids containing polypeptide chains, and similarly omega peptidases that break down the short amino acids, polypeptide chains containing 5 amino acids. Peptidases. There are two forms of peptidases, endo and exo, as we have already seen in the last slide. As we have already discussed about peptidases, there are two forms, endo and exopeptidases. Endopeptidases that act on the internal bond of the polypeptide chain and convert it into smaller peptides, and exopeptidases that act on the terminal bonds of the polypeptide chain. Then comes protein digestion in the small intestine. Small intestine produce different enzymes which are in inactive form and are known as zymogens, that include trypsinogen, chymotrypsinogen, procarboxypeptidases, proelastase, and collagenase. We will study in detail in next slides about these enzymes. Similarly, the release of pancreatic zymogens is mediated by the secretion of two important hormones known as cholecystokinin and secretin that regulate the process of digestion and absorption of the proteins in the small intestine. So, if we look into an overall diagram of an overall mechanism of the enzymes, we find that there are three different organs that help in the digestion process of proteins. So here are proteolytic enzymes of gastrointestinal tract which are secreted by three different organs. That is by stomach. Stomach, apart from HCl, we know that stomach releases two enzymes, pepsin and renin. While pancreas, it releases trypsin, chymotrypsin, elastase, carboxypeptidase 1A and B. And then intestine, along with pancreatic enzymes, intestine also releases its two enzymes, aminopeptidase and dipeptidases. Let's start process of digestion of proteins. If we start from mouth, we know that uh, the oral cavity or the saliva, it produces two enzymes, amylase and lipases. If we have already seen that amylase degrade carbohydrates and lipase degrades fats. So it means there are no enzymes that help to degrade proteins. So the net result is no protein digestion in mouth through enzymes. But unless if we are eating in a raw form, our diet, the first step in digesting, for example, if we take an egg, which is a rich protein diet, or any other solid food, the first step is chewing, and the teeth begin the mechanical breakdown of large diet pieces into the smaller pieces that can be swallowed. So salivary glands then secrete saliva to aid swallowing and the passage of the partially mashed egg through the esophagus. Hence we can say that protein digestion begins from mouth as long as we start chewing. There are two enzymes in saliva that do not help in the protein digestion, but there is mechanical digestion that's of proteins that start from mouth. Then protein digestion in the stomach. If mashed egg pieces enter the stomach from the esophagus, an illustrated image below shows both mechanical and chemical digestion takes place in the stomach. The stomach releases gastric juices that contain hydrochloric acid and an enzyme pepsin. Both of these perform chemical digestion of the proteins. Pepsin initiates the chemical digestion of protein. Actually, there are parietal or oxyntic cells of the gastric gland that secrete HCl, and the pH of the stomach is maintained less than 2. This very high acidic pH of the stomach is responsible for the correct functioning of the pepsin enzyme. The muscular contractions called peristalsis also aid in the process of digestion. The powerful stomach contractions churn the partially digested protein into a more uniform mixture, which is called chyme. Because of the hydrochloric acid in the stomach, it has a very low pH. The range of pH normally falls from 1.5 to 3.5. The acidity of the stomach causes food proteins to denature, unfolding their three-dimensional structure to reveal just the polypeptide chain. This is the first step of chemical digestion of protein, as we can hear in this diagram, that if we recall that the three-dimensional structure of the protein is essential for its proper functioning. So denaturation in the stomach also destroys protein function. This is why proteins such as insulin can't be taken as an oral medication, because its function is destroyed in the digestive tract, first by denaturation and then by further by enzymatic digestion. So instead of taking orally, the sorry the insulin has to be injected so that it is absorbed intact into the bloodstream. The pepsinogen is the inactive form of pepsin that is produced by the gastric cells of the stomach. With the help of HCl, by the action of HCl, the pepsinogen is converted to its active form pepsin. While proteins, by the action of pepsin, they are converted to proteases and peptones. Once proteins are denatured in the stomach, by the action of HCl, the peptide bonds linking amino acids together are more accessible for enzymatic digestion now. And this process is started by pepsin. Now an enzyme that is secreted by the cells. Pepsin begins breakdown of peptide bonds, creating shorter polypeptides. Proteins are large globular proteins, and their chemical breakdown requires time and mixing. Protein digestion in the stomach takes a longer time than carbohydrate digestion, but a shorter time than fat digestion. If we talk about the timings, we can say that and a complete digestion and absorption process of carbohydrate, it take about 2 to 3 hours, while complete digestion of protein, it takes longer time as compared to carbohydrates, that is 4 to 5 hours. While if we talk about fats, it takes about 8 to 9 hours to complete the process of digestion and absorption inside the gastrointestinal tract. So while eating a high protein meal increases the amount of time required to sufficiently break down the meal in the stomach. Food remains in the stomach longer, making you feel full longer and fulfilled. Digestion of protein begins in the stomach, both enzymatically and chemically and mechanically. Sorry, in the sense of chemically actually, because mechanically the digestion of protein starts in mouth. Pepsin is secreted by the chief cells of stomach as inactivated pepsinogen. Pepsinogen is converted to pepsin by removal of 44 amino acids from the N-terminal end by the action of HCl. Pepsin is an endopeptidase and its optimum pH is around 2 to 3. It catalyzes hydrolysis of the bonds formed by carboxyl groups of phenylalanine, tyrosine, tryptophan, and methionine. And by the action of pepsin, proteins are broken down into proteoses and peptones. While here we can see that stomach secretes a strong mineral acid HCl that is very necessary to convert proenzyme pepsinogen towards its pepsin form. While HCl, it denatures protein, it decreases the pH and creates a favorable environment for the functioning of enzyme pepsin, the single enzyme of gastric juice. It activates for protein digestion, it activates pepsinogen and kills some bacteria, so create bacteria-free environment and helps in the absorption of vitamin B12. So at the end of digestion in stomach, protein leaves stomach as mix of insoluble protein, soluble protein, peptides and amino acids in the form of chyme. Pepsin, the functions of pepsin, it acts on peptide bond formed by carboxyl group of phenylalanine, tyrosine, tryptophan and methionine, while it also converts protein into proteoses and peptones. While renin, another enzyme of stomach, it is also called chymosin and is only active in infants and is absent in adult stomach. It performs curdling of milk. It converts casein of milk into paracasein and combines with calcium to form calcium paracaseinate and is only present in infants. Then comes digestion of proteins in the intestine. If I talk about digestion of proteins in the intestine, first of all, the enzyme of gastric juice, that is pepsin, it will be inactivated here. Its activity will be reduced. Why? Because of the basic nature of the small intestine. As we have already seen and discussed that pepsin work best on the pH around 2, while the pH environment of the small intestine is around 8. So here pepsin activity is reduced. While there are further enzymes which are secreted by itself, the lining of the small intestine, and also there are some pancreatic enzymes that help in the digestion in the small intestine. As soon as the chyme leaves the stomach and enters the small intestine, where the majority of protein digestion occur, the pancreas secretes digestive juices into the small intestine, and these contain more enzymes to further break down polypeptides. The two major pancreatic enzymes, which are secreted as proenzymes and have optimum pH 8, the two major enzymes, they are chymotrypsin and trypsin. Trypsin activates other protein digesting enzymes which are called proteases, and together these enzymes break proteins down to tripeptides, dipeptides, and individual amino acids. The cells that line the small intestine release additional enzymes that also contribute to the enzymatic digestion of polypeptides. Here we can see that pancreatic peptides secrete two types of enzymes, exopeptidases and endopeptidases. Exopeptidases, there are examples of exopeptidases, for example, carboxypeptidase A and B, and the examples of endopeptidases include trypsin, chymotrypsin, and elastase. If we talk about hormone stimulation performed in the small intestine, that is the pancreatic juice secretion stimulated by two hormones, majorly by cholecystokinin. What is the role of this hormone during digestion of protein? We have already seen in last lecture that it's a hormone secreted by the eye cells of the upper small intestine in response to fat, protein, and some non-nutrients. For example, chemostat and a peptide neurotransmitter also secreted by neurons of the central and peripheral nervous system also activates the secretion of this enzyme. So cholecystokinin is a gut hormone or gastrointestinal tract hormone that is released after a meal, which helps in the digestion and reduces appetite. Cholecystokinin used to be known as pancreozymin, also its second name given to this enzyme, due to its actions on the pancreas. But now it is commonly abbreviated to CCK. Cholecystokinin is produced by eye cells in the lining of the duodenum and is also released by some neurons in the brain. It acts on two types of receptors found throughout the gut and central nervous system. The most organized functions of this hormone, they are in the digestion and appetite. It improves digestion by slowing down the emptying of stomach of food from the stomach and stimulating the production of bile in the liver, as well as the release of bile from the gallbladder. Bile acts like a detergent, making the fat droplets smaller so that enzymes can break it down more easily. It means that bile act on the large molecules of fats and protein and break it down into smaller molecules so that enzymes can easily break them down further. Cholecystokinin also increases the release of fluid and enzymes from the pancreas to break down fats, proteins, and carbohydrates. So, cholecystokinin seems to be involved with appetite by increasing the sensation of fullness in the short term, that is during a meal rather than between meals. This picture shows the summary of protein digestion. Note that lines representing polypeptide chain in the stomach consists of strings of amino acids connected by the peptide bonds here. Even though the individual amino acids are not shown in this simplified representation, but from stomach, this whole chyme, it enters after the, we can recall here that till after the denaturation by the HCl in the first step, and by the, that is by the hydrochloric acid, and by the enzymatic digestion uh, by pepsin enzyme, this kind enters the small intestine. Here proteins that are not fully digested in the small intestine, they pass into the large intestine. But first of all, we can see that in the small intestine, trypsin, chymotrypsin, and other proteases enzymes, they they break down further these large peptides into smaller peptides, and ultimately in the enterocytes, tri and dipeptides are further broken down into amino acids, and these amino acids, they can be easily absorbed in the bloodstream. So the proteins that are not fully absorbed, digested in the small intestine, they pass into the large intestine and are eventually excreted in the feces. The key enzyme for activation of zymogen is enteropeptidase, produced by intestinal, mostly duodenal cells of the intestine and mucosal epithelial cells. Enteropeptidase, they cleaves off a hexapeptide, that is 6 amino acid fragment from the N-terminal end of trypsinogen to produce trypsin. Trypsin is an active form of the enzyme. Trypsin is a common activator of all other pancreatic zymogens to produce the active proteases, namely chymotrypsin, elastase, and carboxypeptidase. It also acts on the it also hydrolyzes the peptide bond formed by the basic amino acids, that is arginine and lysine. Chymotrypsin, trypsin, and elastase, they are endopeptidases, and active endopeptidase is active at neutral pH of the small intestine. Gastric HCl is neutralized by pancreatic sodium bicarbonate in the intestine and creates favorable pH, that is 8, for the action of proteases. The amino acid serine is essential. As we can see here, the amino acid serine is essential at the active center to bring about the catalysis of all the 3 pancreatic proteases. Hence, these enzymes are referred to as serine proteases, that is trypsin, chymotrypsin, and elastase. Trypsin and chymotrypsin degrade the proteins into small peptides, further hydrolyzed into dipeptides and tripeptides by carboxypeptidases present in the pancreatic juice. The carboxypeptidase is activated by trypsin. They are metalloenzymes, that is requiring zinc for its activation. The pancreatic proteases result in the formation of free amino acids and small peptides, that is 2 to 8 amino acids. There are two forms of carboxypeptidases, that is carboxypeptidases A and B. A, they acts on the carboxy terminal peptide bond that is connected to tyrosine, phenylalanine, or tryptophan. And carboxypeptidases B, they act on the carboxy terminal peptide bond and connected to arginine and lysine. Both of them are form of exopeptidases and require zinc for their activation. Then comes intestinal digestion of proteins. The luminal surface of intestinal epithelial cells, they contain aminopeptidases and dipeptidases. The aminopeptidases is a non-specific exopeptidase that cleaves N-terminal amino acids one by one to produce free amino acids and smaller peptides. Dipeptidases act on different dipeptides to liberate amino acids. There are two examples of amino acids. Aminopeptidases. The leucine aminopeptidase LAP, it releases the N-terminal basic amino acids and glycine, and the second one proline aminopeptidase or prolidase, which it removes proline from the end of polypeptides. Dipeptidases and tripeptidases, they will bring about the complete digestion of protein towards di and tripeptides. Synthesis of the enzymes as inactive precursor protects the exocrine cells from destructive proteolytic attack. The pancreas further protects itself against self-digestion by making a specific inhibitor, a protein which is called pancreatic trypsin inhibitor that effectively prevents premature production of active proteolytic enzymes within the pancreatic cells. The table shows specific enzymes of the digestive tract, gastrointestinal tract, and their appropriate or respective enzymes on which they act upon and perform breakdown of amino acids of these polypeptide chains. Then comes absorption of amino acids. As far as absorption is concerned, it's important to know that, first of all, there are 5 different carriers for amino acids. Actually, the absorption is the process of absorption of proteins is the same as we have already seen in case of carbohydrates. That is, the absorption occur mainly in the small intestine by sodium dependent active transporter system, that is known as sodium amino acid co-transport system, and this is an energy requiring process. Here the di and tripeptides, after being absorbed, are hydrolyzed into free amino acids in the cytosol of epithelial cells. The activates of dipeptides, they are high in these cells. After a protein meal, only the free amino acids are found in the portal vein. So when we talk about absorption process, the carrier is very important. There are 5 different carriers of amino acids. As we know that depending upon the nature and the chemistry of amino acids, there are some amino acids that have acidic nature, some have basic nature, and others are neutral. So same is the case with transporter. There are specific transporter for acidic amino acids, specific transporter for basic amino acids. There are other transporters that are specifically made to carry neutral amino acids, and there is another transporter specific for amino acid, the amino acid number 20, and the transporter for beta amino acids. So if we summarize the whole process till now, starting from dietary proteins, that is by the action of HCl and pepsin in the stomach, these dietary proteins are converted and denatured and partially hydrolyzed proteins. These hydrolyzed and denatured proteins with the action of proteases, they are converted to small peptides and amino acids in small intestine, and then by the process of active transport they are carried towards general bloodstream in the form of amino acids. If we talk about absorption of intact proteins and polypeptides, we can say that short period or immediately after birth, the small intestine of infants and newborns, they can absorb intact proteins and polypeptide by endocytosis or pinocytosis. While intact proteins and polypeptides, they are not absorbed by the adult intestine. Macromolecular protein absorption in certain individuals, there appears to be responsible for antibody formation that often causes food allergy. What happens to the absorbed amino acids? Once the amino acids are in the blood, they are transported to the liver. As with other macronutrients, the liver is the checkpoint for amino acid distribution and any further breakdown of amino acids, which is very less and minimum. Dietary amino acids they then become part of the body's amino acid pool. Now assume that the body has enough and too much glucose and other sources of energy. And so then these amino acids they will be used in one of the following ways. That is, they might be used to synthesize protein in cells around the body. For example, these amino acids might be used to make our hairs, nails, and other body organs or muscles, strengthen the muscles. For example, these amino acids may make non-essential amino acids needed for protein synthesis or may make other nitrogen containing compounds and might rearrange and store as fats. We know that there is no storage form of proteins. So if we have excess number of amino acids, they might be stored as fat in our body, might be converted to fat and stored as in the form of fat. While on the other hand, if we have not enough glucose or energy available resources, so then these amino acids might be used as fuel. So there may be ways like these amino acids may be rearranged into glucose. That is, they may be converted into glucose for fuel for the brain and red blood cells to be used at brain, for example, are these amino acids, they may be metabolized as directly fuel for an immediate source of ATP. And it is also important that amino acids are recycled. Let's start absorption process, the peptides absorption through the small intestine. Starting from lumen, that is the small intestine, the absorption process is the same as we have already seen in case of carbohydrates. That is across the brush border membrane. The absorption takes place with a sodium dependent ATPase pump. Here, instead of potassium, we observe that protein takes place a pump known as sodium hydrogen ATPase dependent pump, where sodium ion, positive ion, is moved along the concentration gradient, that is from higher concentration towards lower concentration, while along with it hydrogen is moved against the concentration gradient. The movement of this pump is carried along with the peptide movement, that is peptide along with its peptide carrier also moves at the same time from lumen of the small intestine towards the enterocytes. Now, what are enterocytes? Enterocytes, they are intestinal absorptive cells. They absorb fats, proteins, and carbohydrates from intestinal lumen. They are simple columnar epithelial cells which line the inner surface of the small and large intestine. A glycocalyx surface coat that contains digestive enzymes. Microvilli on the apical surface increases its surface area. This facilitates transport of numerous small molecules into the enterocyte from the intestinal lumen. It includes broken down proteins, fats, and sugars, as well as also absorption of water, electrolytes, vitamins, and bile salts are carried from with the help of these cells. Enterocytes also have an endocrine role, secreting hormones such as leptin. Because amino acids are building blocks that the body reserve in order to synthesize other proteins, so more than 90% of the protein ingested does not get broken down further than the amino acid monomers. That is, it is carried in the form of amino acids towards the bloodstream. Here is the same diagram that shows transport of peptides and amino acids across the brush border membrane of the small intestine. That is, the transport, the absorption is sodium mediated and sodium ATPase mediated with sodium ATPase pump. We can see that these carrier, there are different types of carrier, some which carry neutral amino acids, some which carry basic amino acid, acidic amino acid, and amino acids, the 20th amino acid. Entrance of some amino acid is via active transport and requires energy. In adults, essentially, it is important that all protein is absorbed as tripeptides, dipeptides, or amino acids. And this process occurs in the duodenum or proximal jejunum of the small intestine. The peptides or amino acids, they pass through the interstitial brush border by facilitative diffusion or active transport. Active transport sodium and ATP to actively transport the molecule through the cell membrane. Once passed through the membrane, the amino acid or peptides, they are released into the intestinal bloodstream and are transported to the liver by the hepatic portal vein. This is known as enterohepatic circulation. In the liver, almost 50% remain and are used to synthesize protein. 50% of amino acids, they are used to synthesize proteins, while nitrogen containing compounds and also to synthesize purine and pyrimidine bases. In some cases, these amino acids may be converted to energy. The liver regulates the amino acid levels in the blood. The amino acids that do not stay in the liver, they pass through and are transported to the rest of the body, to the taken up and utilized by other cells. While in this process, most of the branch chain amino acids that are passed through the liver. In intestine, kidney tubules and brain, the absorption of neutral amino acids is affected by the gamma glutamyl cycle, also known as Meester cycle. The functions of this Meester cycle are peptide and protein synthesis, number one. Number two, it regulates tissue glutathione level. And number three, this cycle transport amino acids across the cell membrane. We can see here that a glutathione molecule, that is a GSH, gamma glutamyl cystinyl glycine, is essential for this Meester cycle. It reacts with the amino acid that is coming from the outside membrane and to form gamma glutamyl amino acid. This gamma glutamyl amino acid is catalyzed by gamma glutamyl transferase. The glutamyl amino acid is then cleaved to give the free amino acid. The net result is the transfer of an amino acid across the membrane. Then the transport of one molecule of amino acid and regeneration of again GSH, the glutathione, requires three molecules of ATP. That is one here, second here, and third here. This is the fate of nitrogen, that is nitrogen metabolism overview. Immediately after birth, the small intestine of infants that can absorb intact proteins and polypeptides, while for adults, the uptake of proteins occur by a process, and in infants, the uptake of protein occur by a process known as endocytosis and pinocytosis. The direct absorption of intact proteins in infants is very important for the transfer of maternal immunoglobulin, that is gamma globulin, to the offspring. While intact proteins and polypeptides, they are not absorbed by the adult intestine. While in adult intestine, the amino acids, they are produced by the polypeptide chains because these amino acids, they contain nitrogen. So if we talk about fate, we can see here that the amino acids coming from dietary proteins or other intracellular proteins, they may be converted to carbon skeletons and amino group. The carbon skeleton further is degraded, enters the citric acid cycle and is degraded to produce carbon dioxide, water to generate ATP, and may be converted into oxaloacetate and might be converted to glucose. While if we talk about the amino group that contain a nitrogen, first it can remain on the molecule and be incorporated into the product that cell is making, for example, a polypeptide. The second fate of the nitrogen may be transaminated. In other words, transaminated is the transfer of amino group from one carbon skeleton to form a new amino acid. For example, if we talk about transfer of amino group from alanine to another amino acid known as to another compound known as alpha glutaric acid to make a glutamic acid. And this process is a water soluble vitamin B6 is needed for this process. The third fate is the deamination process. In this process, the amino group may be removed from the amino acid in a process known as deamination. This process is used for the excretion of the nitrogen and the carbon skeleton is used to produce energy. Again, vitamin B6 is needed for this process. And the fourth one, the nitrogen removed from amino acid is excreted, might be excreted by several different routes, like the most familiar path is urea cycle and is converted to urine or the urea nitrogen excretion product, where most of the nitrogen is in the form of urea here. Nitrogen is also excreted in the feces, skin, hair and nails. While in skin, hair and nails, the nitrogen is bound to protein as this is the building block of each organ. Amino acids are also recycled and may create a pool to form the new proteins. Amino acids from food and those from protein destruction are placed into a pool. Though it is not a literal pool, when an amino acid is required to build another protein, it can be acquired from the additional amino acids that exist within the body. So amino acids are used not only to build proteins but also to build other biological molecules containing nitrogen such as DNA, RNA, and to some extent to produce energy. So it is critical to maintain amino acid levels within this cellular pool by consuming high quality proteins in the diet. or the amino acids needed for building new proteins will be obtained by increasing protein destruction from other tissues within the body, especially from muscles. This amino acid pool is less than 1% of total body protein content. Thus, the body does not store protein as it does with carbohydrates and with fats. For example, carbohydrates stored in the form of glycogen and starch, and lipids are stored in the form of adipose tissues, but proteins are not stored. But rather, only a small quantity of proteins are present in the form of amino acid pool. Amino acids in the cellular pool come from dietary protein and from the destruction of cellular protein. So amino acids in this pool need to be replenished because amino acids are outsourced to make new proteins, energy and other biological molecules. There are some clinical or medical problems that can occur due to malabsorption and maldigestion of proteins. For example, the very important is acute pancreatitis, where premature activation of trypsinogen inside the pancreas itself will start autophagy or autodigestion of pancreatic cells. And the other one is celiac disease, where a malabsorption resulting from immune mediated damage to the small intestine responds to digestion of gluten. So in this case, the gluten is found in the wheat and barley. So in this case, the patient having celiac disease is advised to avoid the diet containing wheat and barley. So our lecture related to digestion and absorption of proteins ended here. Thank you.